Okay, let's unpack this. Um, Imagine you're a first-year medical student, yeah, and you're gearing up to explain something pretty complex like human muscles
Yeah.
to a curious 10-year-old.
Right.
Not just what they do, but how they work, what they're actually made of, and what happens when things go wrong. So our mission in this deep dive is really to give you those aha moments, those simple anchors, so you can make that explanation not just possible, but genuinely like fun and memorable for anyone.
Yeah, and what's truly fascinating here is how we'll journey from the tiniest part, I mean, literally molecules within the muscle cell,
Wow.
all the way up to the big picture, how they help us move, pump blood, even digest our food.
Yeah.
And then, importantly, connect this to what happens when these incredible systems face a challenge. It's really about building that clear mental map.
Absolutely. And muscles, I mean, they really are the unsung heroes, aren't they?
Yeah.
Making up, what, almost half our body mass?
40 to 50%, yeah. Huge proportion.
Constantly generating force, whether you're, running a marathon or just blinking your eyes. And it all boils down to these two key proteins interacting, right? Actin and myosin.
Exactly, actin and myosin. And just to help us navigate this world, maybe remember a couple of linguistic anchors, little shortcuts.
Okay, like what?
When you hear myo, just think muscle, like myalgia, that's muscle pain.
Got it. Myo for muscle.
And when you hear sarco, think flesh, or specifically muscle cell. So sarcoplasm is just the cytoplasm inside a muscle cell.
And sarcolemma.
That's its outer membrane, yeah. These prefixes will be like our little guideposts as we go.
Perfect. Okay, so let's start at the top then. Our bodies have all these different jobs, which means different kinds of muscle. What are these three main types that keep us running?
Great question. Yeah, think of them as three really distinct work teams. First up, you've got skeletal muscle. These are the ones you consciously control, like deciding to flex your bicep.
Okay, the voluntary ones.
Then there's cardiac muscle. This is only in your heart, working away day and night, no conscious thought needed from you.
Right. The hair muscle.
And third, smooth muscle. This is kind of the hidden workforce managing all sorts of automatic internal stuff, like in your gut or blood vessels.
Okay, three teams. To make that even clearer, you use that analogy of work teams. Can you flesh that out a bit? What kind of roles are they playing?
Yeah, let's try this. Imagine three types of construction workers on a big project.
Okay.
Skeletal muscle, they're like the volunteers. You tell them exactly what to do, lift that beam, move this stack. They can lift heavy things fast, but they get tired, and you have to keep giving orders.
Makes sense. Volunteers.
Cardiac muscle, that's your dedicated pump operator. One critical job, keep the water flowing. Works tirelessly, never gets a day off, has its own rhythm. You don't tell it when to pump, it just does.
Always on the job.
And smooth muscle is the behind-the-scenes crew. They're doing all that vital work you barely notice, moving supplies, adjusting ventilation, cleanup, all without direct orders, just quietly keeping things humming along.
That's a fantastic way to visualize it. Okay, let's start with skeletal muscle, then our volunteer team. What are their main jobs, and what really makes them unique?
Right, these are your boss muscles, because you're the boss. They're under somatic control, voluntary control. Their main roles, well, obviously moving your body, holding your posture, keeping joints stable, and even generating heat, helping keep you warm. Thermoregulation.
Ah, interesting. I didn't think about the heat part so much.
Yeah, it's significant. They contract quickly, powerfully, but here's a key thing. They have kind of limited ability to fully repair themselves after a big injury. Cell renewal isn't their strong suit.
And it's not just one size fits all for skeletal muscle, is it? Some are sprinters, some are marathon runners, metaphorically speaking.
Exactly, just like different tools for different jobs. Skeletal muscles have different fiber types. Think of type I fibers as your endurance guys.
Okay.
There are O-red lots of oxygen-carrying myoglobin and slow twitch. They contract slowly but can go for ages. Think postural muscles in your back.
Holding you up all day.
Precisely. Then you have type I IB. These are your power fibers. White, less myoglobin, and fast twitch, built for quick, precise, explosive movements like finger muscles or the muscles moving your eyes.
Wow, okay. Fast ones.
And type TI are sort of intermediate, a blend, found in major leg muscles for things like walking, running, a mix of power and endurance. Understanding this helps explain why different athletes have different muscle builds.
Right, the sprinter versus the marathoner.
Yeah.
Okay, now here's where it gets really cool, especially for our first-year med student listener. How is a big muscle like your bicep actually organized? Down to the tiniest working bit, if you had to explain that to a 10-year-old.
Good challenge. I'd say, imagine a really thick electrical cable. That whole cable, that's your muscle.
Like a big cable.
Inside, you've got smaller bundles, like thinner wires packed together. We call those muscle fascicles.
Fascicles, got it.
Now, pull out one of those thinner wires. That's a muscle fiber. And here's the thing. That fiber is actually one single, really long muscle cell.
One cell. Wow. Okay.
Now, peek inside that single cell, you'd see even tinier threads. Those are myofibrils.
Myofibrils.
Inside the fiber.
Yep. And the fundamental repeating unit that actually does the contracting, the part that physically shortens, that's the sarcomere.
The sarcomere. So that's the tiny engine doing the work.
That's the one, the basic contractile unit.
Can you paint a picture of that little engine? What's its structure like?
Okay, think of the sarcomere like a tiny repeating train car.
A train car. I like it.
Each car has walls at the ends. Those are the Z discs.
This is Z disc. Okay.
Inside the car, you have two main types of filaments. Thin filaments, mostly made of actin, and thick filaments made of myosin.
Actin and myosin again. Right. The thin actin filaments attach to those Z-disc walls and reach inwards. The thick myosin filaments are kind of floating in the middle.
Okay.
Now, when you look at muscle under a microscope, you see stripes, right? Striations.
Yeah, the stripy pattern.
Those stripes come from how these filaments are arranged. The lighter areas, the I band, that's where you only see the thin actin, like the empty space near the doors of the train car.
Light bands, just actin.
The darker areas, the A band, that's where you see the thick myosin, often overlapping with the thin actin. Like the middle of the car with seats and luggage racks, that overlap makes it look darker.
A band, dark, has the myosin. I band, light, just actin. Between Z discs.
Got it.
That train car analogy really works. So how does this tiny train car actually, well, shorten?
Yeah.
To make the muscle contract? That's the sliding filament theory, isn't it?
Exactly. And it's beautifully simple, really. Imagine our thick myosin filaments have little oars or hands sticking out. These are the myosin heads.
Okay, oars on the myosin.
These oars want to grab onto specific spots on the thin actin filaments. Think of them like little Velcro patches on the actin rope.
Velcro patches on the actin. Okay.
Normally, those Velcro patches are covered up. There's a protein rope called tropomyosin lying over them, blocking the myosin oars.
So the oars can't grab the rope because tropomyosin's in the way.
Precisely. It's like a safety cover.
So what removes the cover? What's the magic key?
Calcium. Calcium ions. When your brain sends that signal.
Okay, calcium. Okay.
The muscle cell releases calcium ions. Think of calcium as the magic key. It unlocks a tiny little switch protein on the actin filament called troponin.
Calcium turns the troponin switch.
Yep. And when troponin switches, it pulls that blocking rope, tropomyosin, out of the way, exposing the Velcro patches on the actin.
So now the oars can grab.
Exactly. Now the myosin oars, heads, can finally bind to the actin rope. They form what's called a cross bridge. Then, using energy from ATP,
The cell's fuel.
the oars pull on the actin rope, sliding it inwards. This pulls the Z discs closer together, shortening the whole train car, the sarcomere.
Pulling the ends of the car together.
Right. Then the oar lets go, grabs another patch further down the rope, pulls again. It's like rowing a boat, hand over hand, pulling the rope, making the whole sarcomere shorter and shorter.
That's incredible. Hand over hand rowing, shortening the train car.
Yeah.
So how does the brain electrical signal actually get to these tiny train cars and tell them, okay, start rowing?
Good question. That whole process is called excitation-contraction coupling. It's linking the nerve excitation to the muscle contraction.
Okay.
So a nerve signal arrives at the muscle surface. This triggers an electrical wave, a change in electrical charge called depolarization that spreads across the muscle cell membrane, the sarcolemma.
An electrical wave across the surface.
Then this electrical signal dives down into the muscle cell through little tunnels, and it tells the cell's internal calcium storage tank, the sarcoplasmic reticulum, release the calcium.
And calcium floods out.
And floods onto those actin and myosin filaments. Finds troponin, moves tropomyosin.
And boom, the rowing starts.
You got it. That's the coupling.
Fantastic. Okay, so for our medical student looking through a microscope, how would they know for sure they're looking at skeletal muscle? What are the key visual clues?
All right, histology clues. You'd look for long cylindrical cells, quite large, and crucially, multiple nuclei, the cell's control centers, and they're pushed right out to the edges, under the membrane. Peripheral nuclei.
Multiple nuclei on the edge. Okay.
The inside, the cytoplasm, usually looks pinkish, eosinophilic, and the dead giveaway. Those really clear striations, the stripes, the alternating dark A bands and lighter I bands from our sarcomeres.
The train car batter.
Exactly. If you cut the muscle crossways in transverse section, those striations would look like little dots inside the cell representing the cut ends of the myofibrils.
Got it. Long, stripy peripheral nuclei. What about where the muscle actually attaches to the bone, that connection point?
Right, the myotendinous junction. That's where the force from all those contracting sarcomeres gets transmitted to the tendon, which then pulls the bone.
How does that connection work?
The tendon fibers actually interlock with folds in the muscle cell membrane, the sarcolemma. It's like lacing fingers together, creates a really strong anchor point for transmitting that force.
Makes sense.
Interestingly, though, this junction is often the weakest link in the whole muscle tendon unit. It's a common spot for tears, like in sports injuries.
Ah, okay. Good to know. And you mentioned skeletal muscle doesn't repair perfectly. Are there any backup systems?
Yeah, while the main muscle cells, the fibers, can't divide, there are these special little repair crew cells hiding out on the surface. They're called satellite cells.
Satellite cells.
They're usually quiet, quiescent, but if there's minor damage, they can wake up, divide, fuse together, and help patch up the muscle fiber, or even fuse with existing ones to make them bigger. That's hypertrophy.
So some repair is possible.
Some, yes, but for really significant damage, the body often just fills the gap with fibrous scar tissue, not fully functional new muscle.
Okay. That covers skeletal muscle brilliantly. Let's move to our second work team. Cardiac muscle. The heart's unstoppable pump. What makes it so special?
Well, its primary job is simple but vital. Pump blood, nonstop your whole life.
Right.
The key difference is its control. It's entirely autonomic and intrinsic. You don't think, beat, heart, beat. It sets its own rhythm, though that rhythm can be adjusted, by nerves or hormones.
So it runs itself.
Pretty much. Structurally, unlike those long, straight skeletal muscle cells, cardiac cells are branched. They link up with neighbors. And here's a crucial difference, especially for explaining this simply. Mature heart muscle cells generally cannot renew themselves.
No satellite cells for the heart.
Essentially, no significant regeneration capacity in adults. If there's major damage, like a heart attack, the injured tissue is replaced by fibrous scar tissue. It doesn't grow back as working heart muscle.
Wow. That really underscores why heart attacks are so serious. The damage is permanent scar tissue.
Exactly. That scar tissue doesn't contract, so the heart pump weakens.
So how do all those branched heart cells manage to work together in perfect synchrony, like one giant coordinated pump?
That's where intercalated discs come in. These are really specialized junctions connecting cardiac cells end to end.
Intercalated discs. Okay.
Think of them like super strong, specialized Velcro connectors holding the cells together tightly. But even more importantly, they contain gap junctions.
Gap junctions.
Tiny tunnels, basically, that let electrical signals pass directly and very quickly from one heart cell to the next. It's like they're electrically wired together.
So the signal spreads instantly.
Almost instantly. This allows the whole chamber of the heart to contract as a coordinated unit, a syncytium. They also have strong anchoring points for the actin filaments, called adherens junctions, holding everything together during contraction.
Makes sense. And is there a system coordinating that signal?
Yes, there's a whole conduction system. Part of that involves specialized electrical wire cells called Purkinje fibers. These are modified heart muscle cells, larger and paler than normal ones because they're packed with glycogen.
Purkinje fibers.
You find them within the heart walls, and they help conduct that electrical impulse rapidly and efficiently to ensure a coordinated, effective bump.
Okay, so if we're back at the microscope, how do we spot cardiac muscle? What are the telltale signs?
All right, histology again. You'll see elongated cylindrical cells, yes, but the key is they are branched. That's a big difference from skeletal.
Branched cells.
They usually have just one or maybe two nuclei, and importantly, those nuclei are located in the center of the cell, not pushed to the edge like in skeletal muscle.
Central nuclei, okay.
You will still see striations, that same striped pattern from the sarcomeres, but the absolute clincher, the feature that screams cardiac muscle, is seeing those dark, thick lines where the cells join end-to-end, the intercalated discs.
Branched. Central nuclei, striations, and those intercalated discs.
Got it. You see those discs, you know you're in the heart.
Perfect. And finally, let's look at our third team, the behind-the-scenes crew, smooth muscle. Where do we find them, and what are their quiet but crucial jobs?
Smooth muscle is, well, it's everywhere doing the automatic stuff. Think about your entire digestive tract pushing food along through peristalsis from your esophagus right down to the end.
The gut muscles.
Yep. Also in your respiratory passages, controlling airway diameter, your urinary system, like the bladder wall, reproductive organs, and critically, in the walls of your blood vessels, arteries, veins regulating blood flow and pressure.
So controlling tube diameters, essentially.
A lot of it, yes.
Yeah.
Squeezing things, moving things along. Even the tiny muscles in your iris that change your pupil size, or the little erector pili muscles attached to hair follicles that give you goosebumps.
Goosebumps. That's smooth muscle.
That's smooth muscle contracting, yes. Its control is mostly autonomic, involuntary, often intrinsic to the organ itself, and heavily influenced by hormones and local factors like oxygen levels. Truly automatic.
Wow, they really are everywhere doing hidden work. What about their ability to repair or grow? Are they like skeletal or cardiac?
Here's where they really shine. This is a key difference. Unlike cardiac muscle, smooth muscle cells retain their ability to divide throughout life. They have mitotic activity.
They can divide, so they can regenerate.
Yes, they're excellent at renewal and repair. A fantastic example is the uterus during pregnancy. The smooth muscle wall grows enormously, partly because the existing cells get bigger, hypertrophy, but also because they actually divide and increase in number, hyperplasia.
So the uterus grows by both swelling existing cells and making new ones. That's amazing.
It really is. Shows their adaptability.
They contract using the same actin-myosin rowing mechanism we saw in skeletal and cardiac muscle.
The basic players, actin and myosin, are there, but the mechanism is quite different. They don't have that organized sarcomere structure, which is why they lack striations. They're smooth.
No train cars.
No train cars, no T-tubules or troponin system either. Instead, their actin and myosin filaments are arranged more diagonally, crisscrossing within the cell, and they anchor onto structures called dense bodies, which are like internal anchor points.
Dense bodies instead of Z-discs.
Kind of analogous, yes. So when they contract, the whole cell doesn't just shorten linearly, it actually twists up, sort of like wringing out a dishcloth. It creates this corkscrewing motion.
Corkscrewing, that's different.
Very different. And the contraction itself is much slower, more sustained, perfect for jobs like maintaining blood vessel tone or slowly moving food through the gut. And remarkably, it uses less ATP, less energy than skeletal muscle contraction. Very efficient for long-term work.
Slower, sustained, efficient twisting contraction.
Okay.
And under the microscope, how does our student spot smooth muscle?
Okay, visually. They are typically spindle-shaped cells, thicker in the middle, tapering at the ends, like a stretched-out football shape.
Spindle-shaped.
They have a single, centrally located nucleus, which often looks elongated, almost cigar-shaped when the cell is relaxed.
Single central nucleus.
And critically, no striations. That's the big giveaway. No stripes at all. They're generally smaller than skeletal muscle cells and often arranged in sheets or bundles rather than distinct fibers.
And if they're contracted?
If they're contracted, the whole cell looks shorter and fatter, and that central nucleus might even look squished or twisted, reflecting that corkscrew action.
Okay. Spindle shape, single central nucleus, no stripes. Got it. Are there any variations?
Yeah, there are a few specialized types, like myoepithelial cells, star-shaped cells that wrap around glands, like salivary or mammary glands, and help squeeze out secretions, or myofibroblasts, which appear during wound healing. They're like fibroblasts that can also contract, helping to pull the wound edges together.
Interesting specializations. Okay, so we've met our 3 muscle teams, incredible systems. But what happens when things go wrong? When they get injured or maybe just aren't used enough?
Right. This brings us to muscle repair and remodeling. It's important to remember your muscles aren't static. They're constantly being rebuilt, broken down, adapted, like a house always under renovation.
A continuous process. And does each work team handle repairs the same way? Or does it depend on the muscle type?
That's a crucial point, and connected back to what we said earlier, the answer is definitely no. It's very different across the 3 types.
Okay, let's recap the repair abilities.
All right, skeletal muscle. The main cells, the fibers, can't divide, but remember those satellite cells?
Or repair crew.
Exactly. They can divide and fuse to patch things up, leading to repair or growth, hypertrophy. But for big injuries, it often ends up as fibrous scar tissue, not perfect replacement.
Okay, limited but some repair via satellite cells. What about cardiac?
Cardiac muscle, in adults, essentially no regeneration. Injury, like a heart attack, leads to fibroblasts coming in and laying down scar tissue. The damage is largely permanent, replaced by non-contractile tissue.
No repair crew there. Very vulnerable.
Very.
Yeah.
And then smooth muscle. These guys are the champions of repair. They keep your ability to divide, mitotic activity. They can form new smooth muscle cells, increasing in size, hypertrophy, and number, hyperplasia, like we saw with the uterus.
Smooth muscle can actually make more smooth muscle cells.
Okay. Yep, much better at regeneration.
Beyond sudden injury, what about the changes from just using muscles more or less, the remodeling.
Right, that's remodeling, and it's always happening. For skeletal muscle, believe it or not, the main contractile proteins, actin and myosin, are completely replaced about every 2 weeks.
Every 2 weeks? Wow.
Yeah, constant turnover. So, hypertrophy. If you exercise regularly, the building up of these proteins outpaces the breakdown. More myofibrils, more mitochondria, the muscle gets bigger and stronger, like adding more bricks and stronger beams to your house.
Use it and build it up.
Exactly, atrophy. But if breakdown outpaces replacement, the muscle wastes away. This can be disuse atrophy if you're sedentary or a limb is in a cast. The fibers shrink, you lose power, like a house crumbling from neglect.
Use it or lose it.
Pretty much. Then there's sarcopenia, which is specifically age-related muscle loss. It can start as early as your 30s, and by 80, you might have lost half your muscle mass. More fat moves in between the fibers.
Age-related wasting. Sarcopenia.
And finally, denervation atrophy. If the nerve supply to a muscle is cut or damaged, the muscle can't get signals to contract. It weakens and wastes away rapidly. It's like the power cable to the house is completely severed. The muscle is essentially useless without the nerve.
That makes sense. Can nerves repair, though, to restore function?
Sometimes, yes. In the peripheral nervous system, if the main nerve cell body is okay, the cut axon can try to regrow, guided by supporting Schwann cells. If it successfully reconnects with the muscle at the neuromuscular junction, function can be restored, but it's a slow process.
Okay, so there's hope sometimes after nerve injury. Now, let's shift gears and delve into some specific problems of muscle disorders. What happens when the signals get blocked or the muscle structure itself fails?
Right, looking at pathologies. And just to set the stage, let's clarify a few terms quickly. A myopathy is any primary disease of the muscle itself.
Okay, muscle disease.
Dystrophy specifically refers to degeneration, usually from a genetic cause. Atrophy is wasting, like we just discussed. Hypertrophy is enlargement by bigger cells. Hypoplasia is enlargement by more cells, mainly in smooth muscle. And sarcopenia is that age-related wasting.
Got the terms. Okay, first up, myasthenia gravis. You often hear about this causing muscles to tire out really easily.
That's the hallmark symptom, yes. Myasthenia gravis is an autoimmune disorder. Your own immune system gets confused and attacks healthy tissue.
Attacks what, specifically?
It attacks the receptors for the neurotransmitter acetylcholine, ACH, at the neuromuscular junction, the point where nerve tells muscle what to do.
So the muscle's mailbox is for the nerve's message.
Exactly. Imagine the nerve sends the AC message, but antibodies created by the immune system are like gum stuck in the mailbox. They block the AC from binding or even destroy the mailboxes altogether.
So the message just doesn't get delivered properly.
Right. Fewer signals get through, so the muscle contraction is weaker. The structure of the junction itself can also degrade, making transmission less efficient.
And what does that feel like for the person?
The main thing is fatigability. Muscles work okay at first, but weaken rapidly with repeated use. Common signs are drooping eyelids, mitosis, double vision, slurred speech, difficulty swallowing, and in severe cases, breathing problems. It often gets worse with stress or tiredness.
Who tends to get this?
It's most common in women under 40 and men over 60, but it can occur at other ages too.
And how do doctors manage it? Is there a cure?
There's no cure, but it's manageable. Avoiding triggers is important. Medications called acetylcholinesterase inhibitors are key. They stop the enzyme that normally breaks down ACE.
Letting the message hang around longer.
Exactly. Gives the ACE more time to find one of the working mailboxes. Sometimes removing the thymus gland helps, as it can be involved in producing those faulty antibodies.
Okay, that makes sense. What about other problems at that nerve-muscle junction? You mentioned botulism and organophosphate poisoning.
Yeah, they affect the same junction, but in opposite ways. Botulism toxin from the bacteria prevents the nerve from releasing ACE in the first place.
So no message sent at all.
Right. Leads to paralysis, muscle weakness. Organophosphate poisoning, like from certain pesticides or nerve agents, does the opposite. It blocks the enzyme that breaks down AA.
So you get way too much AA.
Way too much. A massive buildup, constantly stimulating the muscle and nerves. Leads to symptoms like sweating, tearing, seizures, muscle twitching, paralysis, and a respiratory failure. Complete overload.
Two extremes affecting the same junction. Okay, let's move away from the junction and into the muscle itself. Genetic disorders, the muscular dystrophies. You mentioned a case earlier, a 15-year-old boy illustrating Duchenne muscular dystrophy, DMD.
That's right, classic presentation. Started with leg weakness around age 4, progressively worsened, wheelchair-dependent by 12, developed scoliosis. His blood tests showed sky-high creatine kinase, a marker of muscle breakdown.
And the key finding?
Complete absence of a crucial protein called dystrophin. DMD is an X-linked genetic disorder, so it primarily affects boys, caused by mutations that stop the body from making functional dystrophin.
Okay, what is dystrophin? Why is it so vital? What does it do?
Think of dystrophin as a critical structural support beam inside the muscle cell. It acts like a shock absorber and links the muscle cell's internal skeleton, the cytoskeleton, specifically actin filaments,
Wow.
to the outer membrane, the sarcolemma, and even connects indirectly to the stuff outside the cell. It basically stabilizes the muscle cell membrane during contraction and relaxation.
So it's like the main support beam holding the house wall, the cell membrane, together during the stress of movement.
Exactly. In DMD, that support beam is missing or completely broken. So every time the muscle contracts, the cell membrane is fragile, unstable, it gets tiny tears.
Constant damage with every movement.
Yes. This leads to calcium leaking inappropriately, inflammation, cell death, necrosis, and gradual loss of muscle fibers, which then get replaced by fat and fibrous connective tissue.
Which explains the weakness. What are the typical signs a doctor or parent might see in a boy with DMD?
Progressive muscle weakness, especially in the legs and pelvis first. You often see calf muscle pseudohypertrophy. The calves look large, but it's mostly fat and scar tissue, not strong muscle.
False enlargement.
Right. A classic sign is the Gower's sign or Gower's maneuver. The child has to use their hands and arms to walk up their own body to stand up from the floor because their hip and thigh muscles are too weak.
Using arms to climb up their legs.
Yep. Also fatigue, maybe walking on toes, a waddling gait, difficulty climbing stairs, frequent falls. Later, breathing problems and heart problems often develop, as cardiac muscle can also be affected. Scoliosis is common, too.
And the outlook.
It's the most common severe form. Incidence is about 1 in 3,500 to 5,000 male births. Life expectancy has improved, but it's still significantly reduced, often into the late 20s or early 30s, usually due to respiratory or cardiac complications. About 2/3 of cases are inherited, 1/3 are new spontaneous mutations.
Such a devastating disease. Is Becker muscular dystrophy related?
Yes, Becker is also caused by mutations in the dystrophin gene, but it's generally milder. In Becker, some dystrophin is usually made, but it's maybe shorter or misshapen, so it still offers some support. Symptoms appear later, progress more slowly. Limb-girdle muscular dystrophy is actually a group of disorders affecting other proteins in that same stabilizing complex around dystrophin.
Okay, different parts of the support structure are failing. Finally, let's quickly cover 2 other important, often acute muscle problems: malignant hyperthermia and rhabdomyolysis.
Right. Malignant hyperthermia, often called anesthesia-related hyperthermia, is a rare but potentially lethal reaction to certain general anesthetic agents or muscle relaxants. It happens in people with a specific genetic predisposition, often a mutation in a gene called RYR1, which controls a calcium channel in muscle.
So it's triggered by anesthesia in susceptible people.
Exactly. When they get the triggering drug, there's an uncontrolled massive release of calcium ions from storage inside the muscle cells.
Like opening the floodgates for calcium.
Totally. Imagine the thermostat controlling the muscle's furnace. Calcium release gets stuck on rocks. This causes massive, sustained muscle contraction all over the body.
What does that lead to?
The muscles become rigid. They generate enormous amounts of heat, hyperthermia, consume huge amounts of oxygen, hypoxemia, and produce excessive carbon dioxide, hypercapnia. The rapid muscle breakdown, rhabdomyolysis, releases potassium, hyperkalemia, and muscle proteins into the blood, and the body becomes very acidic, acidosis. It's a life-threatening emergency.
Wow. And rhabdomyolysis, you mentioned it as part of MH, but it can happen on its own too.
Absolutely. Rhabdomyolysis just means rapid breakdown of skeletal muscle tissue. It can be caused by severe trauma like crush injuries, extreme overexertion, drug abuse, certain infections, or even as a side effect of some medications like statins in rare cases.
So muscle just breaks down rapidly.
Yes. And when muscle cells break open, their contents spill into the bloodstream. A key component is myoglobin, the oxygen-carrying protein in muscle.
Myoglobin leaks out. Is that dangerous?
It can be very dangerous for the kidneys. The large myoglobin molecules can clog up the kidney's filtering system, potentially leading to acute kidney injury or failure. A classic sign is dark, reddish-brown urine, often described as tea-colored. due to the myoglobin being excreted.
Tea-colored urine. A warning sign for kidney damage from muscle breakdown.
Exactly. Needs urgent medical attention.
Phew. Okay, what an incredible journey through the world of muscles. We've gone from those tiny actin and myosin filaments rowing their roads.
That's train cars.
The train cars.
Yeah.
All the way to understanding how complex disorders can affect movement and health. Hopefully for everyone listening, especially our first-year med student, you now have those anchors, those analogies.
The volunteers, the pump operator, the behind-the-scenes crew.
Exactly. To help explain these complex ideas, whether it's for an exam or just to that curious 10-year-old.
Yeah, and if we connect it all back, it really does highlight that intricate dance between structure and function. Understanding the basics, having those simple pictures in your head, it's just fundamental to appreciating how amazing, how resilient, but also sometimes how vulnerable our body's movers really are.
Absolutely. So to leave you with something to think about, building on today's deep dive, consider your smooth muscles. What silent, continuous actions are they performing for you right now, completely unnoticed? And how does their unique, slow, sustained, twisting contraction make them perfectly suited for those vital hidden tasks?
A great question. Keeps those curious muscles working, thinking about the ones you don't think about.
Indeed. Thanks so much for joining us on this deep dive. Until next time.